Hello everyone, the title of the project is DeeReCT APA Prediction of Alternative Polyadenylation Site Usage through Deep Learning. Polyadenylation is a process at the end of transcription in eukaryotic cells, in which a strand of adenine nucleotides is added to the newly formed RNA to terminate the process. This requires enzymatic recognition of a polyadenylation site or path, upstream sequence element, and downstream sequence elements in the mRNA sequence. As it is shown in the left figure, a gene may have multiple polyadenylation sites, and there is a certain probability that each of them will be used, and a gene at each time can only use one polyadenylation site. The goal of this research is to predict the usage of each polyadenylation site of a gene based on its sequence context. Here we introduce some related works of this topic. Published in 2018, the paper entitled Inference of the Human Polyadenylation Code uses a convolutional sequence model to predict the relative strengths of two competing paths. In 2019, another model, DeePasta, is published to compare the relative strength of paths based on genomic sequence and their RNA secondary structure. Both of them have limitations because, first, they only consider two paths at a time, but in reality, a gene may have multiple paths. Secondly, they only give qualitative comparison results of the two paths without giving a quantitative usage estimation of them. Here we introduce DeeReCT APA, Deep Regulatory Code and Tools for Alternative Polyadenylation, which quantitatively predicts the usage of all alternative paths at the time of a given gene. The network architecture is shown in the left figure, where there are n 1D CNN networks, which serve as the base networks to extract sequence features for n polyadenylation sites of a given gene. And there are bidirectional LSTM blocks, which serves as interaction layers to integrate information across multiple paths and handle the problem of different numbers of paths for differ' genes. Finally, the model predicts the percentage of usage of each path, and this model is trained and supervised by 3' region extraction and deep sequencing data as ground truth. This slide summarizes several results of our DeeReCT APA model. In the left table, it shows the performance of DeeReCT APA on BL and SP hybrid mouse 3' reads dataset. On both the parental dataset as well as on the F1 dataset, DeeReCT APA outperforms other models in terms of mean absolute error, comparison accuracy, highest usage prediction accuracy, as well as average Spearman's correlation. In the right figure, through the prediction of GSRR, it shows the benefit of modeling all paths jointly, because only the DeeReCT APA model can predict the relative strength of the two alleles, BL and SP, correctly compared to the ground truth usage. Compared with polyadenylation code, DeeReCT APA also shows better correlation with ground truth in terms of the prediction of allelic usage difference of the BL and SP stream mouse. The conclusion is that simultaneously predicting the usage of all competing paths of a gene is possible with a CNN-LSTM network. Secondly, joint modeling of all competing paths of a gene can improve the performance of the model and capture the information of interactions between the polyadenylation sites.